Hello, this is a video on SARS-CoV-2 and the disease it causes, COVID-19. Please read the disclaimer. You may like to stop the video while reading the disclaimer, and having read the disclaimer, you may like to continue to watch the video. Please note, I am not a medically qualified doctor. No advice is being given. It is not intended to give any advice. This video is for educational purposes only. This is a presentation of literature search and search on other sources such as the internet and my interpretation thereof. These ideas are limited as these have not been peer reviewed. Scientific ideas can change and information can become out of date. And I shall be discussing some autopsy specimen histology slides and so on during this talk. Aims and objectives: to discuss some aspects of COVID-19 histopathology, especially the expression of host and viral cell surface markers. Target audience: university life sciences undergraduates and others with relevant prior knowledge. My name is Harsh Sharma. I have a BSc, MSc, PGCE, and I did a second BSc. This is a list of the things I shall be discussing in this video. I think I've got most of the things listed here. Maybe some things are missing. SARS-CoV-2 is the coronavirus responsible for causing the disease COVID-19. This disease has many manifestations and outcomes. In various slides and discussions in this video, each slide should be taken as a particular manifestation in a particular setting. However, some manifestations are more common than others. This is a structure of the mouse hepatitis virus to illustrate some features of coronavirus. In the middle, there is a ribonucleoprotein containing the RNA and nucleoprotein surrounded by a lipid bilayer membrane, that is, with proteins in the bilayer membrane. This is the membrane glycoprotein, envelope and the spike protein. This is a general diagram of coronavirus. This talk is about SARS-CoV-2 virus, and in particular, please note that SARS-CoV-2 does not possess hemagglutinin esterase. SARS-CoV-2 utilizes ACE2 as a receptor to gain entry to the cell by cell fusion pathway or endocytic pathway. ACE2 is the angiotensin converting enzyme 2. This is a diagram showing the pathways the virus can utilize to enter a cell. This is the endocytic pathway, and that is virus is taken in by endocytosis, endosome is formed, and from there the viral RNA is released into the cell by the help of enzyme cathepsin L. Cathepsin L is provided by the cell. This is the membrane fusion pathway. The virus and the cell membranes fuse, and opening is created through which RNA enters the cell. This is the main method of cell infection in lungs, probably the only method of cell infection in lungs. That is to say, infection of the lungs usually takes place by cell fusion pathway, cell and the virus fusion pathway, leading to insertion of viral RNA into the cell. SARS-CoV-2 binds the cell via the ACE2 and its viral spike protein. So ACE2 is the cellular receptor for the virus, and virus binds ACE2 via the spike protein. TMPRSS2 provided by the cell then cleaves the spike protein. Further events follow, which leads to the fusion of the viral membrane and the cellular membrane. An opening is created to allow the viral RNA to enter into the cell. Main method of cell infection in lungs, and probably the only one, is by fusion of viral and cell membrane, leading to insertion of viral RNA into the cell. This shows the viral entry into the cell via the endocytic pathway. The virus attaches the cell via the ACE2 receptor on the cell and the spike protein on the virus. Virus is taken into the cell via endocytosis, an endosome is formed, and by the action of enzyme cathepsin L, which is provided by the cell, viral RNA is released into the cell. Hydroxychloroquine may not work because the endocytic pathway is not utilized by the virus to infect the cells in the lungs. For the virus to infect the cell, the target cell must provide ACE2 for virus cell attachment and TMPRSS2 for spike protein cleavage for virus entry to the cell. There is also a further requirement that cell provides furin. This happens at a later stage after the virus has gone through its replication and is ready to be released from the cell. And at this stage, the furin cleaves the spike protein. So the spike protein is cleaved at two sites, firstly by furin before the virus is released from the cell, and then by TMPRSS, spike protein is cleaved for virus entry to the new cell. This is a section of the lung, and again another section of the lung. This has been stained for surfactant, that means the Pneumocyte2. So these cells shown by the red arrow are Pneumocyte2. This is the alveolar space and the epithelium here. In this section there is the alveolar space, and it has been stained for ACE2 positive cells. So the brown staining shows cells which are ACE2 positive. Notice very few cells are shown to express ACE2 in this section. And in particular, notice that the staining is polar. It is not all over the cell. It is on certain areas of the cell. When you look at it, if you imagine this is the alveolus, imagine this is a three-dimensional space, and then you can see the surface of the Pneumocyte2 that is stained is the one that is pointing into the alveolar air space. So the surface pointing away from the alveolar air space is not stained. Again, surface of the Pneumocyte2 that is pointing into the alveolar air space is actually stained for ACE2. Alveolar regions had uncommon to regional polarized apical staining of ACE2 on solitary epithelial cells. I will deal with the apical, what apical means later on, but for the meantime, just a reminder that cells are polarized as far as their staining is concerned, and there are very few of them that are actually stained for ACE2. Only 1.2% of Pneumocyte2 express ACE2, so very few Pneumocyte2 are expressing ACE2 on their surface. And those ACE2 expressing pneumocytes 2, so the pneumocyte 2 that actually express ACE2 are ACE2 positive, are larger compared to the ACE2 negative pneumocytes 2, the type 2 pneumocytes. Co-expression of ACE2 and TMPRSS2 on surface of pneumocyte 2. ACE2 is the cell surface molecule to which the virus spike protein binds, and TMPRSS2 is the enzyme on the cell surface which cleaves the viral spike protein. These are serial sections of the tissue. So these slides are made of serial sections of the tissue, and on this slide simply nuclei have been stained, so you can see the nuclei here. On this slide only ACE2 has been stained, and you can see the ACE2 is polarized. And on this slide TMPRSS2 has been stained. And then these three slides have been merged to produce a superimposed section, and we see that ACE2 and TMPRSS2 are found on the same area of the cell. They are co-expressed. Okay, so they're expressed on the same surface of the cell, the same part of the cell. They are both polar in a similar way. Only 1.2% of type 2 pneumocytes express ACE2 on the apical surface, and then only half of the ACE2 expressing pneumocyte 2 also express TMPRSS2. So this represents ACE2 positive cells. Only half are TMPRSS2 positive, and the other half are TMPRSS2 negative. When we do the calculation, 1.2% to start with, only 50% are going to express TMPRSS2. That means in health, only about 0.6% of class 2 pneumocytes are ACE2 positive and TMPRSS2 positive. So there are only 0.6% that are double positive. So in health, only about 0.6% of the class 2 pneumocytes are double positive for ACE and TMPRSS2. Remember, there is also a requirement of furin. Furin is the enzyme that also cleaves spike protein, but as part of the maturation process in the producer cell. A question that arises: the target cells, that is, type 2 pneumocytes that are ACE2 positive and TMPRSS2 positive in the lung for SARS-CoV-2 are very few in number. So with such a small number of cells that the SARS-CoV-2 can actually infect, why does SARS-CoV-2 cause so much damage in the lung? So we need to understand the general anatomy of the respiratory system. So this is the nasal cavity, the pharynx, the trachea, the bronchus, and lungs, alveoli. We will see there are different types of cells in each part of the lung, and we will also see which type does the virus infect, and if the mode of infection is different in these cells from each other. And we will also see that conjunctival epithelial of the eye is also a target cell for the SARS-CoV-2 to infect. One thing that happened is, in a study, different results were obtained with ACE2 expression with two different antibodies. So two different antibodies gave two different results when they were looking for ACE2 expression in the tissues. This is a slide which I shall discuss. It is broken down in the next slide. So this is the histological sections of various tissues in the respiratory tract. So here we have nasal mucosa from different patients. For example, male 58-year-old, male 64-year-old. So these are different histological sections taken from different patients, and they're stained with either of the two antibodies. So there's the antibody MAB933 and there's the antibody HPA000288. Both are antibodies directed against ACE2. So notice in this section, HPA000288 stains for ACE2, but from the same patient, MAB933 does not stain for ACE2. And the amount of staining is different in different patients. And this is in the nasal mucosa ciliated epithelium. So in the nasal mucosa ciliated epithelium, HPA000288 stains for ACE2, whereas MAB933 does not stain for ACE2. In the bronchus ciliated epithelium, again, HPA000288 stains quite clearly for ACE2, whereas MAB933 again is lacking reactivity against ACE2. But when we look at ACE2 in the alveolar cells, we find both MAB933 and HPA000288, they both stain for ACE2. But note, rare ACE2 staining was present in a few alveolar cells. And we will see what we mean by rare ACE2 in the next slide. Out of 360 samples, only 2 samples were found to be positive for ACE2 with HPA000288, and with MAB933 only 1 out of 360 samples was found to be positive for ACE2. In the nasal mucosa, 6 positives and 6 negatives with HPA00288, but with MAB933 all negatives for ACE2. And you can also see results for bronchus, 2 positives and 6 negatives here and 8 negatives here. So MAB933 rare 1 out of 360 samples, ACE2 staining was present in a few alveolar cells. With HPA000288, 2 out of 360 samples, ACE2 staining was present again in very few alveolar cells. Question: is there a bias in reporting? Are negative results not being reported and fewer instances of ACE positive Pneumocyte2 findings are being reported? Are there a variety of antibodies with varying reliability? So, very few cells express ACE2 in the normal lung, but so many cells lost and so much damage. So many clusters of large numbers of virally infected cells, that is by SARS-CoV-2. So take a look, very few cells which are ACE2 positive, but here we have large clusters of cells which are infected by the virus. They are identified as Pneumocyte2. The white staining is for a marker for Pneumocyte2. And the question arises, how can virus affect so many of these cells when there are only few cells in the sample? But remember, this is a disease sample, this is several things are happening here, as we will see. We saw this cluster of cells in the previous slide, and here is another cluster of cells infected by the virus. In this situation, green equals viral RNA. So here it is, lots of cells being infected by the virus when under normal circumstances ACE2 expression is very low, and ACE2 and TMPRSS2 expression is even lower still. One important aspect of a virus in such large numbers is that either the antibody response is overwhelmed, there just aren't enough antibody molecules to combat such a viral load, or the amount of antibody antigen load generated would lead to massive sequelae such as complement fixation, chemotaxis, etc. Proliferation phase of diffuse alveolar damage characterized by proliferation of type 2 pneumocytes. So here is a pneumocyte which is proliferating, replicating after diffuse alveolar damage. This is a diseased tissue, and notice the epithelial layer of pneumocytes 1 and 2 is no longer in its place. So that epithelial layer has come off and it has now been effectively lost completely. Here are some type 1 pneumocytes, disquamated type 1 pneumocytes, and so on. Compare that to the normal lung. Nice airspace, epithelial layer here. This is the epithelial layer for this side, this alveolus. This is the epithelial layer for this alveolus. And notice there is no epithelial layer in this alveolus. Heavily congested capillaries, yes. And there it is, type 2 pneumocyte replicating. So these type 2 pneumocytes have arisen by proliferation of type 2 pneumocytes after damage done by SARS-CoV-2. So in this disease tissue, these type 2 pneumocytes have arisen after proliferation of type 2 pneumocytes. In this slide, there's a massive hyperplasia of the type 2 pneumocytes. These are type 2 pneumocytes that have arisen as a consequence of type 2 pneumocytes replicating. Take a note, this is not SARS-CoV-2 infected lung, but this is infected by SARS, the earlier virus. And in slice B, as the repair occurs, hyperplastic type 2 pneumocytes grow over bare alveolar walls. So it is walls that have been, from which the type 2 pneumocytes and type 1 pneumocytes have been shredded, taken off. Now there are type 2 pneumocytes coming over to grow over to that wall. Why did some pneumocyte type 2 survive and were not infected by the virus? Possibilities include some cells originated from another stem cell without surface expression of ACE2. Some pneumocyte 2 did not express surface ACE2. Some pneumocyte type 2 did not express surface TMPRSS2. Now we come to the asymmetric expression of ACE2 on pneumocytes. Remember, ACE2 is expressed on this part of the cell, this part of the cell that is protruding into the alveolar space. Reminder, ACE2 protein expression on a cell is asymmetric. Very low number of type 2 pneumocytes express ACE2. Even fewer numbers of type 2 pneumocytes co-express ACE2 and TMPRSS2. And reminder that ACE2 and TMPRSS2 are co-expressed. Here we are, a structure of the alveolus showing alveolar type 2 cell. Notice this cell has different morphology to this end to this end. Here, the type that is facing the inner surface, the alveolar airspace, tubular myelin is being secreted, that is the surfactant. So this layer is called the basolateral membrane, and this layer towards the airspace is called the apical membrane. So remember, we were talking about apical site. Here it is, the apical membrane of the class 2 pneumocyte. So reminder that there is the staining of the class 2 pneumocytes protruding into the airspace, alveolar airspace, and here it is, the side that faces the airspace, and this is where you get the high concentration of ACE2 on the apical membrane. So alveolar regions had uncommon to regional polarized apical staining of ACE2 on solitary epithelial cells. So here we are. And also a reminder, TMPRSS2 expression on Pneumocyte2 is also asymmetric. ACE2 and TMPRSS2 co-localized. So now our diagram has progressed to show that high concentration of ACE2 on apical membrane as well as TMPRSS2 is also present on apical membrane here. Present on apical membrane. Now look at SARS-CoV-2 expression on Pneumocyte2. Brown equals SARS-CoV-2 nucleocapsid protein. So this is the slide showing SARS-CoV-2 expression on Pneumocyte2. If you look carefully, you'll see SARS-CoV-2 expressed on this side of the cell, but not on this side. Here's another cell. SARS-CoV-2 is present on this side of the cell, but not on this side. And if you look at this multinuclear giant cell again, SARS-CoV-2 is present on this side of the cell. SARS-CoV-2 expression on Pneumocyte2 is asymmetric. Note that pneumocytes are no longer in correct orientation with respect to alveolar structure. So the alveolar structure has been disrupted. Pneumocytes are no longer where they should be, in the orientation they should be, but we can see that SARS-CoV-2 expression is asymmetric. This is a slide with more detailed labeling, but please note this is my interpretation and expert opinion may differ. So as we said earlier, SARS-CoV-2 expression asymmetrically on this side of the membrane but not on this side. Alveolar airspace here. Some more cells showing asymmetric SARS-CoV-2 antigen. What is happening here? Is this a cell dividing? One cell is SARS-CoV-2 positive and another cell is negative, or it could be something else. Notice SARS-CoV-2 on one side of this multinucleate cell. Notice a prominent nucleolus in this nucleus. So our diagram now becomes high concentration of ACE2 on apical membrane, TMPRSS2 expressed on apical membrane, SARS-CoV-2 enters and released from Pneumocyte2 on apical membrane. So these three things are happening on the apical membrane. Question: is this release of virus at apical membrane leading to increased infectivity of type 2 pneumocytes and increased transmissibility? That is to say, this is going to the airways, the bronchi, trachea, and the nasopharynx in the nose, leading to outside world. Here are virus particles being released from the cell. Does SARS-CoV-2 have strategy to ensure its spike protein does not bind ACE2 on cells producing the virus? Pre-release and post-release of the virus from the cell. So the virus is being released. It doesn't really want to bind to the same cell. that will be counterproductive for the virus. So does it have a strategy that it doesn't bind to the same cell from which it is being produced? So here is SARS-CoV-2 being expressed in the alveoli. This is brown. In this section, the brown shows SARS-CoV-2. In this section, there is a change of color scheme. Red equals SARS-CoV-2, whereas brown is the TTF1, a Pneumocyte2 marker. Here it is in ciliated cells. In the trachea, SARS-CoV-2 antigen being expressed. So up here again brown shows SARS-CoV-2 antigen, whereas up here color scheme has been changed. This is SARS negative. In this slide, SARS-CoV-2 was red. So we are looking for red color showing SARS-CoV-2 antigen. Okay, so these two slides have different color scheme. Earlier on, we discussed this sample and we showed that SARS-CoV-2 was asymmetrically expressed on type 2 pneumocytes. So remember, there it is SARS-CoV-2 on this surface, but not on this surface here. Here it is SARS-CoV-2 absent on this side, but present on this side. But up here we have diffuse staining of SARS-CoV-2 antigen. So what could be the reasons for this diffuse pattern of staining of type 2 pneumocytes by SARS-CoV-2? One, it could be bona fide diffuse staining. So it could be that in sometimes SARS-CoV-2 does infect all of the type 2 pneumocytes and cause diffuse pattern. In others, as we saw, SARS-CoV-2 infection may be asymmetric in the cell. Secondly, overstaining. Too much stain, overspillage in non-SARS containing areas. 3. Tissue degeneration, autolysis. 4. Orientation. If looking directly at apical membranes, SARS-CoV-2 staining would appear all around the cell. So it may be a simple effect of which angle you look at it from. So here we have tracheal epithelium showing infected and non-infected cells. So here it is infected cells staining red and non-infected cells. Same with RNA for viral spike protein in tracheal epithelium. Some cells have viral RNA, others don't. So why are some cells infected and not others? If you look at ACE2 expression in non-infected trachea, again it is sparse. In this experiment, tissue sections are taken from bronchus, lung, and conjunctiva and cultured ex vivo, and then they are challenged by the virus. One must be aware of a limitation of this, that is, the virus is not subjected to various controls of the body, such as the immune system. Nevertheless, it does show tissues which are vulnerable to infection by the virus. And so here it is SARS-CoV-2, SARS-CoV, MERS-CoV, H1N1. And it becomes quite obvious that SARS-CoV-2 does infect bronchus quite heavily. There is varying degrees of infection by the other viruses. H1N1 is the strain they've used is H1N1 PDM. Lungs are also infected by these viruses. In conjunctiva, notice that SARS-CoV-2 infects conjunctiva, but SARS does not. The authors did not have enough material to do experiments with MERS-CoV and H1N1 with conjunctival tissue, so they did not have enough tissue for that. The other caution you have to take is that SARS-CoV-2 has a wide variety and levels of infectivity and severity. A single case may not be representative of a wide population. So again showing the ex vivo infection of conjunctiva by SARS-CoV-2. These are the in vitro cultures of human explant tissues. That is to say, the tissue sections are taken from the patient and cultured in the petri dish and so on, and they are explants so that they maintain the organ architecture. They are not cell cultures, so the organ architecture is maintained. Nevertheless, we have to be careful because certain controls that do exist within the body are not present in the explant situation. For example, these are not subjected to the normal immune regulation. And here are the arrows showing the color of the arrow and the type of cell mentioned, and we also have to be a bit more careful there because we do really need a immunohistological confirmation that the cell type is as mentioned. Nevertheless, these do look morphologically like type 1 pneumocytes showing virus antigen present within the type 1 pneumocytes, that is to say the nucleocapsid protein. Up here the alveolar macrophages, and we have to be even more careful here whether the virus is replicating within the macrophage or it is actually engulfed material. And these do look like type 2 pneumocytes containing the virus antigen. So this is a histologic lung findings at autopsy. So this is from a patient tissue section, and there is detection of SARS-CoV-2 in lungs and airways of patients with COVID-19. Brown equals SARS-CoV-2 nucleocapsid. So there it is, SARS-CoV-2 antigen present. This is a timeline of some patients who died of COVID-19. And the thing to note is, all of these patients had tested positive for SARS-CoV-2. And at death, you got patients 1 to 5 which are positive for SARS-CoV-2 in lungs, but 3 of these are negative in the trachea and 2 are positive in the trachea. And you got 2 patients that are negative for SARS-CoV-2 in trachea and lungs. So what could be happening in these cases is that in some cases is the virus doing a hit and run. That is, virus has been cleared, but the pathological sequelae continued and the patient died. We need sequential SARS-CoV-2 positivity data. Secondly, RT-PCR is more sensitive than histology, and RT-PCR positive samples may be histologically negative. This is a diagram of the nasal cavity, and there is the nasal cavity and nasopharynx, because we will be looking at some samples from this area. Detection of SARS-CoV-2 in nasopharyngeal swab cell preparations, and they were looking for RNA. Panel A is the cases near the onset of symptoms, and B, also cases near the onset of symptoms, shows degenerative changes. You can see the cells are not very healthy. Now, this slide was prepared 2 weeks after the initial swabs. So after the initial swabs, they took further swabs 2 weeks later, and you see the cells are quite healthy. The SARS-CoV-2 much lower load. So panel F shows some SARS-CoV-2 infected cells with lower viral load, that is 2 weeks after the initial swabs. And 4 weeks after the initial swabs, the cells are quite healthy and they've been stained for ACE2, and ACE2 is present on the apical membrane. This is the same slide in panel F on the previous slide, and you can see SARS-CoV-2. And the question here becomes, how are these cells limiting the viral load? So when you compare with the initial onset, the viral load was much heavier, but here 2 weeks later, the viral load is reduced in the cells. Despite the virus infection, these cells look quite healthy. And how are these cells limiting the viral load? This is earlier picture of bronchial epithelium showing SARS-CoV-2 infection, but lots of green ACE2 material on the cell. So we have seen in vivo and ex vivo infection of lung tissue by the SARS-CoV-2 virus. We have seen nasopharyngeal cells, tracheal cells, bronchial epithelium, type 1 pneumocytes, type 2 pneumocytes, and endothelial cells being infected by SARS-CoV-2. So now we come to some pathological aspects. As we saw, high concentration of ACE2 and TMPRSS2 on apical membrane, and the virus is released from the apical membrane. One of the things that leads to is pathological change. Immune cells are coming in, damage takes place, and the alveolus is filled up with protein-rich fluid. This protein-rich fluid dries up to form hyaline membranes, which can thicken. There it is. This septa has now thickened due to immune cell infiltration. Next two cases, and we see asymptomatic patients. Coincidental findings of COVID-19 as part of patient's cancer management. So this patient was being managed for cancer as part of the tumor resection. They took some normal tissue away from the lung, and few days later this patient then showed signs and symptoms of COVID-19. There was nothing there with this patient to show, no symptoms indicating this patient had SARS-CoV-2 and COVID-19 disease. Look at this. Proteinaceous exudate in the alveolar airspace. Protein globules in the alveolar space. Balls of fibrin. Intraalveolar fibrin with early organization. Inflammatory cells and multinuclear giant cells. Hyperplastic pneumocytes. So this is a biopsy from a patient. This patient was asymptomatic at the time of biopsy. 4 days after the biopsy, patient developed symptoms. Resection of the right middle lobe was carried out to remove tumor, and section away from the tumor was removed for this histological section. This case presented in early phase of pandemic. Although CT scan showed findings indicative of SARS-CoV-2 at that time, these findings were not associated with SARS-CoV-2. The other thing to note is you got several different types of pathological changes occurring in the patient's lung concomitantly. So this is happening, this is happening, this is happening in the lung at the same time. And also note that the hyaline membranes have not yet formed. Another patient, asymptomatic, coincidental finding of COVID-19 as part of patients' cancer management. In A, evident proteinaceous and fibrin exudate. So there it is, proteinaceous and fibrin exudate. In B, diffuse expansion of alveolar walls in septa owing to fibroblastic proliferation in type 2 pneumocyte hyperplasia. So here it is. Look at the septa are now thickened. C, plugs of proliferating fibroblast balls in the interstitium. So here it is, plugs of fibroblasts there. And D, abundant macrophages infiltrating air spaces in type 2 pneumocyte hyperplasia. So again, notice different changes at the same time. So these patterns of histopathology are occurring in the patient's lung concomitantly. Again, hyaline membranes have not yet formed. So hyaline membranes happen at a later stage in the pathological sequelae of this. This is a slide taken from Adventures in Neuropathology. A very nice talk, and notice this part of the lung is relatively normal, and here we have pathological changes taking place. So in a given patient, different parts of the lung may be in different stages of lung pathology. In this slide, please note slides are from different patients presented to illustrate a possible scenario. Magnifications are not standardized. So here we have the fluid is dried up to form hyaline membranes. If you look in the hyaline membrane, this thread is SARS-CoV-2 antigen, virus spike protein in hyaline membrane. That's red. So that's the viral spike protein in hyaline membrane. So this is the RNA in hyaline membrane, SARS-CoV-2 RNA. So a possible scenario is normal lung, nice air spaces, a layer of epithelial cells for this alveolus, a layer of epithelial cells for this alveolus, protein globules, certain pathological changes taking place, hyaline membranes form. These hyaline membranes contain virus particles. So here we are clearly showing hyaline membranes with SARS-CoV-2 antigen, hyaline membranes with RNA, viral RNA. And in this slide it shows that there is an immune cell infiltration, fibroblast infiltration to cause thickening of the septa. This is extra pinky material here, more pink, that's fibrin. We have to ask, why is there an infiltration of immune cells that is causing septal thickening? Are there things left over in the lung which are now contained within hyaline membranes and causing this immune infiltration and other aspects of pathology? Nice staining here, SARS-CoV-2 RNA within hyaline membranes. In this one, immunohistochemistry for SARS-CoV-2 nucleocapsid antigen in hyaline membrane. We have to consider what else is included in this protein-rich fluid in the alveolar space. Possibilities: virus remnants, Pneumocyte2 remnants with cytokines within the Pneumocyte2, for example IL-6 and other interleukins in the fluid phase, antibodies, complement components, and the list goes on and on. So all this dries up to form hyaline membranes. So all of this can dry up to form hyaline membranes. And then These can act as inflammatory mediators, leading to inflammation and thickening of the septa and so on. Does hypertension force more fluid out of the capillary into the alveolar space? It is now generally accepted that hypertension is one of the comorbidity factors for COVID-19. So a question is being asked, if the person has hypertension, that means the blood arrives at a higher pressure at the lung capillary. And one of the problems COVID-19 creates is edema developing in the alveolar space. So higher pressure would tend to, is it possible that higher blood pressure is forcing more fluid out of the capillary into the alveolar space? So the question is being asked, does higher pressure force more fluid out of the capillary into the air space and create more edema? Different cytokines are produced by SARS-CoV and CoV-2 virally infected Pneumocyte2 cell line A549. So if you take cell line A549 infected by SARS-CoV, the cells will now produce this set of cytokines. If you take the same cell line A549 infected with CoV-2 the cells will now produce this set of cytokines. So different viruses have different signature of cytokine production by the cell they actually infect. And this is the immunoblot of the cytokines produced by two different viruses by the same cell line, infected by the same cell line. Infection of cell line A549 either by SARS-CoV or CoV-2 leads to production of different cytokines. The cytokine profile produced by pneumocytes in response to a given virus may vary in different individuals. This may be one contributor of varying pathology. Cytokine profile of virally infected individuals can be affected by other parameters such as virus to cell ratio. Cytokine profile of virally infected individuals may depend on circumstances at a given time, such as other concurrent infection. Now returning to the issue of very few cells that are staining for ACE2 in the alveolar epithelium, yet large numbers of virally infected pneumocytes, clusters of virally infected pneumocytes, and see what's going on there. So there is a missing link. We got to try to explain how it is very few normal type 2 pneumocytes express ACE2, and then there are a large number of type 2 pneumocytes in clusters that are virally infected. A possibility is that on many cells there is sufficient ACE2 on the cell surface for SARS to infect the cell, but the actual amount of ACE2 is low and below the detection limit. Other cytokines may induce expression of ACE2 in Pneumocyte2. Proliferation of cells may be a signal for ACE2 expression. SARS-CoV-2 can utilize another surface molecule for infection. We need also to consider, is there another aspect of COVID-19 pathology that increases ACE2 expression? So these are possibilities. All these are possibilities, nothing proven, but things that may be happening. This slide shows infection with SARS-CoV-2 increases ACE2 expression in nasal swabs. So COVID-19 patients, mRNA for ACE2 is much higher compared with control. Again, you look at it here, very few ACE2 cells positive, but look at this SARS-CoV-2 infected bronchus epithelium. There's a lot of ACE2. How is that? We were saying that ACE2 is sparse on in the lungs as expression of ACE2, but here it is SARS-CoV-2 infected bronchial epithelium, loss of ACE2 expression here. ACE2 is green. The other thing is we may well have two disease processes, in fact more than two disease processes. One, infection of pneumocytes, two, infection of endothelial cells. So there is infected pneumocytes in the lung, here is infected endothelial cells. So SARS-CoV-2 in the lung, endothelial cells, here it is. Quite clear staining of the endothelial cells by SARS-CoV-2. This is again a slide taken from Adventures in Neuropathology and very clearly marked perivascular infiltration of immune cells. So there's a blood vessel here, around it a collection of immune cells. Blood vessel, around it collection of immune cells. So there is again very clearly perivascular immune cell infiltrate. This is taken from another paper, thrombosis of two medium-sized pulmonary blood vessels with marked endothelitis, fibrinoid necrosis of the vessel wall, and thrombi in engorged surrounding capillaries. So here it is, again, infiltration, perivascular infiltration of immune cells. Please note that these slides are from different patients and they have not been standardized for magnification. There is this perivascular immune cell infiltrate around the vessel, and the question that I want to ask is it possible that there's endothelial cell infection going on here which is attracting an immune response? So is it possible that, like here, there is an endothelial cell infection by the virus, that there is actually an infection by the virus that is causing an immune response, plus other sequelae? We come to complement fixation. It's outside the scope of this talk to discuss complement in detail, but just suffice to say C4D is a mark of a complement having been fixed. It is a high stable covalent bond and it remains bound as a footprint while antibodies dissociate over time. So wherever there has been complement fixation, C4D remains there as a footprint of that complement fixation has taken place. And if you look here in endothelium in this slide, there is clearly C4D deposition here. That is to say, the complement has been, there is evidence that there was complement fixation here. In this slide, it shows very clear C4D deposition. So in green here is C4D, showing that complement fixation has taken place. Here is SARS-CoV-2 spike protein, showing that there has been infection of the endothelium. Superimpose the two together and you see C4D and SARS-CoV-2 are co-expressed. That is to say, they are C4D is being deposited where there has been SARS-CoV-2 infection. If you look at this electron microscope, this is a pneumocyte cytoplasm. In fact, Pneumocyte2, that's a big clue here. It is a Pneumocyte2 lamellar body surfactant, and you can see virus particles coming from the cell. So the cell looks quite healthy and it's giving off virus particles. And up here the cells also look quite healthy. They're infected by the virus, but they are reasonably healthy cells. Here is a comparison of different cytopathic effects of the virus, different outcomes of infection by the virus on the cell. So here we have cell lysis. The virus actually lyses the cell completely, and here we have cells are in pretty good healthy state. Although here the architecture is destroyed, but the cells look reasonably healthy. Here the cells look reasonably healthy. The endothelium is reasonably intact. So, and here the cells are degenerating. So the virus can have different outcomes depending on which cell is infecting under what circumstances. This slide in a bit more detail. Cell lysis here. Look at these cells. They are about to lyse. These cells have lysed here already. These cells are weakened, and they will lyse soon. And some more already happened. Cell lysis here. Remember this slide. Cells are reasonably healthy and positive for viral nucleoprotein. Cytopathic effects of SARS-CoV-2. We are looking at gross cytopathic effects, and we must remember, although gross cytopathic effects are not seen, viruses hijacking cell machinery and causing unwanted change in cellular function. So, when we look at 16-HBE cells, there is no cytopathic effect caused by SARS-CoV-2 virus, although these cells are subject to cytopathic effects by other viruses. On the other hand, vero cells undergo cytopathic effects after challenge with SARS-CoV-2 virus. Here they are, 16-HBE cells growing well in culture, and there is the graphs showing viral titer. So these cells, although quite healthy, growing in culture, they are releasing SARS-CoV-2 into the medium. They are infected by SARS-CoV-2, and SARS-CoV-2 is being released. And when you assay for SARS-CoV-2, firstly, simply looking at the viral titer in the supernatant, you can see for about 7 days there is virus production going on. When the assay is carried out by plaque assay, looking at the infectivity of the virus of new cells, again you can see there's a lot of virus being produced until about day 6 or 7. So here cells, 16-HBE cells are growing in culture. They have been stained for virus nucleoprotein, so these cells are infected by the virus, and they seem reasonably well despite the fact that they are infected by SARS-CoV-2. This staining is for the nucleus, and here we have merging of these two pictures, and you can see the nucleoprotein, virus nucleoprotein, N protein is around the nuclei, so it's quite healthy cells. And they're quite healthy cells here, and you can see virus SARS-CoV-2 being extruded from the cell at the electron microscope level. So here is the virus coming out of the cell. The cell looks reasonably healthy, and the virus particles are pouring out by, I would guess, by exocytosis or a similar process. So here it is, virus particles coming out. Again, the cell looks reasonably healthy. So the virus is utilizing this method of replication, whereby it's exiting the cell without killing the cell off. No cell lysis, just continuous exclusion of virus particles, we can see. So SARS-CoV-2 may be growing differently in different cells within human body. Lytic or non-lytic, induction of cytokines, induction of interferons, response to cytokines or interferons. All these things may vary from one person to another. We should have some caution in interpreting cell lysis. Infection may be in early stages, not yet reached the point of cell lysis. Lysis may be due to immune-mediated cell damage. There may be cell autolysis independent of viral effects. Nevertheless, it's worth mentioning that a few slides ago, we actually saw viruses coming out of cells that were intact and virus was being released. Right, now we come to an important topic. Is ACE2 an interferon stimulated genes? Firstly, I did not find any data showing or implicating that ACE2 expression in type 2 pneumocytes is interferon regulated. There is data showing ACE2 expression is increased by interferons in some cell types in airways, but not alveoli. So, is ACE2 an interferon stimulated gene? Some evidence says yes, ACE2 is a type 1 interferon stimulated gene, and ACE2 is also type 3 interferon stimulated gene. On the other hand, no. There's a paper that says interferons and viruses induce a novel primate-specific isoform of the ACE2 and not the SARS-CoV-2 receptor ACE2. But I'm not aware of this data being published in a peer-reviewed journal or confirmed by other workers at the time of producing this video. When we look at the question of is ACE2 an interferon stimulated gene, there are two sides of the story, two complexities. One, we need to look at types of interferons, and two, we need to look at the different cells of the lungs and the airways, types of cells that are responsive to interferon. Interferon is produced by a cell that is infected by the virus. So virus enters the cell, it is recognized as a foreign intruder, and one of the response of the cell is to produce interferon. So this interferon now leaves the cell and binds to receptors on interferon receptors on neighboring cells. In response to this, the cell then synthesizes antiviral proteins which help to protect the cell against virus attack. So interferon is produced by a cell that is infected by the virus and is released to protect cells nearby. There are three main types of interferons: type 1, type 2, and type 3. Type 2 interferon is part of the immune response against the virus. It's produced by immune cells, and we will not be dealing this in detail as it is part of an immune response. Type 1 interferon interferes with virus reproduction in cell. Type 1 interferon alpha is produced predominantly by virus-infected leukocytes. Type 1 interferon beta can be produced by most nucleated cells, especially fibroblasts. Type 1 interferon receptors are found on most cells. Type 3 interferons interfere with virus reproduction in cell. Type 3 interferon receptors are largely restricted to cells with high susceptibility to pathogenic encounters, such as the cells that line our intestine and lungs. Type 3 interferon group is a group of antiviral cytokines that consists of 4 interferon lambda molecules called interferon lambda 1, 2, 3, and interferon lambda 4. Viral infection induces upregulation of interferon lambda, mRNA in epithelial cells, peripheral blood mononuclear cells, and dendritic cells. So, type 1 or type 3 interferon is produced depending on the cell type. Some cells will produce type 1 predominantly, some cells will produce type 3 predominantly. Interferon induces ACE2 in keratinocytes. So if you look at keratinocytes, no interferon, interferon alpha, interferon gamma, there is ACE2 induction. What we must remember is interferon alpha is the one we discussed. Interferon gamma is produced by immune cells. Is interferon net beneficial or detrimental in SARS-CoV-2? Viral infection leads to bystander epithelial and/or immune antiviral response. Increase in interferon that causes increase in interferon-stimulated gene expression, and interferon acts on other cells to express interferon-stimulated genes. In their study, this work was done with interferon alpha. This is the reference. And the authors say, our study finds that type 1 interferons and to a lesser extent type 2 interferons upregulate ACE2. This is based on several lines of evidence. 1. We identified a human goblet secretory cell subset in upper airway nasal epithelium enriched for ACE2 expression to have the highest interferon alpha induced gene signature. 2. We found that interferon alpha and to a lesser extent interferon beta or interferon gamma induced ACE2 expression in a published data set of air-liquid interface cultures derived from human nasal epithelial cells. So interference, possible outcomes. 1. Interferon is not produced, maybe virus has evading mechanisms. Number 2. Normal amount of interferon is produced, limiting virus propagation and helping specific immune response to develop. Number 3. Too much interferon and or prolonged interferon production leading to damage carried out by interferon. 4. Interferon produces effects favoring viral replication, e.g. ACE2 upregulation and more cells expressing ACE2 on cell surface. But in this case, note, cells will also need to express TMPRSS2 as well as furin. So on the whole, looking at the data on interferon, I personally think a lot more clarification is needed. Further experimental work needs to be done to really clarify the situation about the role of interferon in ACE2 stimulation. This paper has not yet been published in a peer-reviewed journal. Nevertheless, we need to consider the contents of this paper. And what is being presented is that a new isoform of ACE2 is identified, delta ACE2. Delta ACE2 does not function as ACE2, no carboxypeptidase activity, and does not bind SARS-CoV-2 spike protein. Okay, so this is the thing we need to consider as well, whether the antibodies are binding delta ACE2 and not ACE2 and so on. So there's a fair amount of confusion that needs to be resolved, I believe. In my opinion, the data needs to be a bit more clearer on the role of ACE2 and the expression of ACE2 in cells. Now we look at the question of more or less ACE2. Number of uninfected type 2 pneumocytes expressing ACE2 may be increased. This benefits the virus and provides more target cells. But again, reminding, TMPRSS2 also needs to increase. I do not have data to support this, but there is a possibility that TMPRSS2 from a cell may help to cleave spike protein in an adjacent cell. But it is suspected ACE2 expression in the individual infected cell may be decreased or downregulated. Further suggestion: this may benefit the virus. Newly synthesized virus does not bind the ACE2 via its receptor until released from and attached to another cell. This may also be a reason for having cleavage buffurin held back until it is ready to leave the infected cell. Also, when a virus has infected a cell, it stops other new virus particles binding to the cell so that these particles go on to infect other cells. Furthermore, it stops other virus particles from entering its own occupied cell. So, on one hand, increase in ACE2 on cell surface will allow more viruses to attack the cell. On the other hand, the cell that is infected by the virus already, it doesn't want more ACE2, it wants less ACE2 receptors so that other viruses do not attach and bind to the cell. And also, the virus does not want to bind to ACE2 within the cell before it's released. That is the strategy the virus would use for its own benefit. So factors may lead to increase in ACE2, increase in number of pneumocytes, hyperplasia, interferon-induced increase in ACE2, unknown viral effects. Factors may lead to decrease in ACE2, virus causing destruction of Pneumocyte2. So if cells are destroyed that could express ACE2, then there is going to be reduction in ACE2. Virus induced decrease in ACE2, that is synthesis, interference, and so on. And thirdly, masking of ACE2 by SARS-CoV spike protein. So spike protein simply binds ACE2, so it cannot easily be detected. Possible effects on ACE2 activity and other components by Pneumocyte2 cell death and hyperproliferation. So we have a certain number of Pneumocyte2 in the lung. Virus infection kills cells, and it's reasonable to assume that it will kill preferentially ACE2 positive cells. So the total number of cells will decrease, and on top of that, the proportion of ACE2 positive cells will decrease also even more. So, and there will be a decrease of other factors such as the surfactant cytokines. Then there will be hyperproliferation. This, assuming the proportion of new ACE2 positive cells is the same as original population, then total ACE2 will increase and may cause problems by increased ACE2 activity. So here ACE2 activity decreases as compared to normal physiological activity because total number of cells are less, as well as the proportion of ACE2 positive cells is less also. Here the total number of cells are increasing and ACE2 positive cells will increase also. So there will be an increase of other factors also, including surfactant cytokines and so on. And so there may be a problem by increased ACE2 plus and increase of other factors, and up here there may be a problem by decrease of ACE2 plus and decrease in other factors. What are the effects of increased ACE2? Total expression on general physiology other than SARS-CoV-2 aspects. So total increase in ACE2 activity may be due to increased expression on a cell or increase in number of cells expressing ACE2. Expression of ACE2 on pre-existing known ACE2 cells or new cells produced by cell division that also express ACE2. What are the effects of increase in total ACE2 expression? Total increase may be due to increased expression on a cell, increase in number of cells expressing ACE2. This could be increase in pre-existing non-ACE2 expressing cells expressing ACE2, or new cells are produced that also express ACE2. So this will lead to increased total ACE2 activity, including from here. So there are several arms leading to the increase in total ACE2 activity. And up here, there will be increase in number of target cells for SARS-CoV-2 in fact. So increase in pre-existing non-ACE2 expressing cells to express ACE2 and new cells produced that also express ACE2. This will lead to increase in number of target cells for SARS-CoV-2 to infect, and also added on to that increased expression on a cell, there will be increased total ACE2 activity. What are the possible effects of increased ACE2? Total expression on general physiology other than SARS-CoV-2 aspects. Briefly, angiotensinogen is converted to angiotensin 1 by renin, and ACE causes angiotensin 1 to angiotensin 2, and each of these is further acted upon by ACE2, so that there is angiotensin 1 to 9 and angiotensin 1 to 7, which then goes on to become angiotensin 1 to 5. So, if there is a decrease in ACE2, this pathway is blocked, so you get a buildup of angiotensin 2, and that will lead to increase in vasoconstriction, increase in hypertrophy, increase in fibrosis, increase in proliferation, increase in atherosclerosis, decrease in natriuresis, and decrease in diuresis. So if you have increase in ACE2, then this pathway will take place. Angiotensin 1 is converted to angiotensin 1 to 9, angiotensin 2 is converted to angiotensin 1 to 7, and you will get these effects taking place. So, possible effects of increase in ACE2 levels. So if you have a increase in pneumocytes by hyperplasia, then it's logical to assume there'll be more cells expressing ACE2 on the surface, so the total amount of ACE within the individual will increase, and this will lead to this pathway being favored, that is decrease in vasoconstriction, decrease in hypertrophy, decrease in fibrosis, decrease in proliferation, decrease in atherosclerosis, increase in natriuresis, and increase in diuresis. A decrease in ACE2 due to loss of pneumocytes, 2 due to SARS-CoV infection. If the SARS-CoV-2 infection destroys Pneumocyte2, then there is going to be a decrease in ACE2 because it's got rid of ACE2 expressing cells.